Good afternoon, all. So today we can discuss about culture methods. That means different types of culture methods. First of all, we can discuss aerobic culture methods. So, giving an introduction, growing bacteria in pure culture is still one of the most widely used methods in microbiology. Many bacteria, particularly those that cause diseases and those that are used in scientific studies such as research, are heterotrophic, which means they depend upon organic compounds for their food to provide energy and carbon to them. So, some bacteria also require added nutritional components such as vitamins in their diet. An appropriate physical environment must be created where important factors such as temperature, pH, and the concentration of atmospheric gases, particularly oxygen, are controlled and maintained. So, bacteriological culture medium can be prepared as liquid broth, solid, which can be either plate media or slant media, or as semi-solid. So these portions we have discussed. Solid and semi-solid media contain a solidifying agent called agar or gelatin. Agar, it is a polysaccharide derived from red seaweed, Rhodophyceae, and it is preferred because it is an inert and non-nutritive substance. The agar provides a solid growth surface for the bacteria upon which the bacteria are able to reproduce until the distinctive lumps of cells that we call the colonies form. So these all factors you know. Next is growing bacteria in culture. A population of the bacteria which are grown in the laboratory. It is termed as culture. The culture can be a pure culture which contains only one single type of organism. Then a mixed culture which contain two or more different types of bacteria. If the bacterial culture it is left in the same media for too long, the cells use up the available nutrients and they start excreting toxic metabolites, and eventually what happens? The entire population will die. Thus, the bacterial cultures, there is a need of the bacterial cultures to be periodically transferred to the new media. That method is called subculturing to keep the bacterial population viable and growing. Otherwise, if we are keeping the bacteria in that same media, this utilization of the available nutrients becomes more and it starts to excrete toxic metabolites which eventually kill the bacteria or the microorganisms. Microbiologists use subculturing techniques to grow and to maintain the bacterial cultures and also to examine the cultures for purity or morphology or to determine the number of viable organisms. In clinical laboratories, subculturing is used to obtain a pure culture of an infectious agent and also for studies leading to the identification of pathogen. These bacteria can live almost anywhere. Subculturing steps must be performed aseptically to ensure that unwanted bacterial or fungal contamination is kept out of an important culture. So the bacteria should be maintained in aseptic condition. And so the subculturing should be performed aseptically to prevent the unwanted growth of other microorganisms. A microbiological culture or microbial culture, it is a method of multiplying microbial organisms by letting them to reproduce in a predetermined culture media under controlled laboratory conditions. Microbial cultures are foundational and basic diagnostic methods used as a research tool in molecular biology. Then next is microbial cultures are used to determine the type of the organisms, the abundance in the sample being tested, or both. So the microbial cultures determine which type of organism and the abundance of samples. It is one of the primary diagnostic methods of microbiology, and it is used as a tool to determine the cause of infectious disease, and by letting the agent multiply in a predetermined medium. For example, if we are taking a throat culture by taking the scrapings from the lining of the tissue in the back of the throat and blotting that sample onto a medium to be able to screen for the harmful microorganisms such as Streptococcus pyogenes, which is the causative agent of strep throat. Furthermore, the term culture, it is more generally used informally to refer to selectively growing a specific kind of microorganisms in the lab. It is often essential to isolate a pure culture of microorganisms. So, there is a necessity of isolating pure culture. What do you mean by pure culture? Pure or axonic culture is a population of cells or multicellular organisms growing in the absence of other species or types. So, in the absence of other species or types, a pure culture may originate from a single cell or a single organism, in which case the cells are genetic colonies of one another. So, it should be the organisms present in one pure culture should be identical, should be different from other organisms. For the purpose of gelling, the microbial culture, the medium of the agarose gel or agar is used. So for gelling, for solidifying the microbial culture, we are using either agar or agarose gel. Agar is a gelatinous substance derived from seaweed chip that we know that guar gum, which can be used to isolate and maintain the thermophils. Next is about bacterial culture. There are several different types of bacterial culture methods that are selected based on the agent being cultured and the downstream use. Broth culture. One method of the bacterial culture is liquid culture, in which the desired bacteria are suspended in a liquid nutrient medium. This may allow to grow up large amount of bacteria for a variety of downstream applications. Liquid cultures are ideal for preparation of antimicrobial assay, in which experimenter inoculates liquid media with bacteria and let it grow overnight. So in broth culture, it is this method may allow us to grow up large amount of bacteria for a variety of downstream application. So the liquid cultures are mainly ideal for the preparation of antimicrobial assay, and in which the liquid broth is inoculated with the bacterium and it is allowed to grow overnight. By whether we can use in a shaker to perform a uniform growth. Then they would take aliquots of the sample to test for the antimicrobial activity of a specific drug or protein. So liquid cultures can be used for antimicrobial assays. Next is about as an alternative, the microbiologists may decide to use static liquid cultures also instead of shaking. These cultures are not shaken and they may provide microorganisms with an oxygen gradient. So sometimes they are either using shaker or they are keeping it as a static. Next is about agar plates. Agar plates, in this the microbial The microbiological cultures can be grown in petri dishes. Now in the liquid culture, it is simply inoculating the organism into the broth or liquid media. In agar plates, the microbiological cultures can be grown in petri dishes of different sizes that have a thin layer of agarose based growth medium. So this type of medium, it is present with agar. Once the growth medium in the petri dish is inoculated with any type of microorganism or desired bacteria, the plates are kept for incubation at optimal temperature for growing a specific type of a selected bacteria. For example, usually at 37 degrees Celsius or the human body temperature. For the cultures from humans or animals, or lower for environmental cultures. So after this incubation or desired level of growth is achieved, agar plates can be stored upside down in a refrigerator. Now it can be stored in a refrigerator for an extended period of time to keep the bacteria for future experiments. In order to maintain the viability, we are keeping it in refrigerator. There are a variety of additives that can be added to agar before it is poured into plate and allowed to solidify. So we can add different types of additives. Some type of bacteria can grow only in the presence of certain additives. This can also be used when creating engineered strains of bacteria that contains antibiotic resistance genes. When the selected antibiotic is added to the agar, only the bacterial cells containing the gene insert conferring resistance will be able to grow. So when the selected antibiotic is added, the bacterial cells which contain the genes which confer the resistance that are able to grow in the antibiotic added media. And this allows the researcher to select only the colonies that were successfully transformed. The very important method in the microbiology is to isolate a single type of bacteria from a source that contain many. So the most effective way to isolate a single bacteria from a group, that is streak plate method, which dilutes the individual cells by spreading them over the surface of an agar plate. Single cells reproduce and create millions and millions of clones, which all pile up on the top of the original cell. The pile of these bacterial cells observed after this incubation period are called colonies. Each colony represents descendants of a single bacterial cell, and therefore all the colonies are clones. Therefore, you can transfer a single colony from a streak plate to new media. You have achieved a pure culture with only one type of bacteria. So you can transfer a single colony from the streak plate. Thus, we can isolate individual colony. Next is about streak plate method. So we are coming to the streak plate method. To obtain a pure culture, it is required to obtain a separate single bacterial colony. And for attaining this, a well-known method, that is a quadrant streaking method, can be used. There are other methods also. Now I am mentioning here quadrant streaking method. Here the microbial mixture, it is seeded on the edge of the agar plate. I will show the picture with an inoculating loop and then streaked out over the surface. After seeding the culture, the first sector is streaked originating from the seeding area. After the first sector is streaked, inoculum loop is sterilized and the second sector is streaked using the inoculum from the first sector, like that. There are, I will show the picture.